Hey everybody, I'm going to talk a little bit about my project I've been working up using isotopically labeled pyruvate as a precursor for sparse isotopic labeling of a large protein, in this specific case, the toxoplasma skip1 protein. Large proteins struggle with spectral resolution because they tumble slower in solution, leading to more spin-spin interactions and faster relaxation times, causing increased line width within a spectrum. As protein size increases, the likelihood of resonance overlap also increases, which can lead to frequency degeneracies and spectral crowding within the spectrum. So we can combat this using two techniques that we are going to implement using sparse isotopic labeling of the methyl group along with deuteration. Isotopic labeling of the methyl group uh, enhances spectral resolution, as if only the methyl group is labeled, we would have three equivalent protons contributing to a signal resonance in the absence of coupling to other isotopically labeled carbons, since only the methyl would be labeled. And these methyl groups would have altered relaxation properties that can give us sharper signals. And deuterated species are often already used to improve resolution in NMR by eliminating proton contributions to spin relaxation. But in our case, we would be using partial deuteration, making a species with two hydrogen atoms and one deuterium. So it would make the methyl carbon behave more like a methylene that can show a sharp triplet peak caused by the canceled alpha and beta proton moments. So how exactly can we achieve sparse labeling on a large protein? Amino acid labeling has been done in the past, oftentimes with supplementing specific labeled amino acids, like alanine in our case is what we would want. Specific labeled amino acids in combination with other substrates that suppress scrambling to other amino acids. These isotopically labeled versions of these higher level substrates are often quite expensive, and they may not offer the possibility of exchanging methyl deuterons for methyl protons. So what we could do, and what is often done, is that media is supplemented with alpha keto precursors or citric acid cycle precursors like glucose, and it's much more cost effective and leaves possibility for proton deuteron exchange using the host's own enzymes. This also lets us study the host metabolism as well, and the effects of isotopic compounds on the host. So an experiment similar to the second method I explained, glucose, is often used for experiments like this. However, glucose has its limitations. When going through glycolysis, if we were to provide the medium with isotopically labeled glucose at the carbon 1, it goes through glycolysis and isomerizes and cleaves into two pyruvate molecules. So the pyruvate methyl, one of them would come from C1 and one of them would come from C6. Only one of them would be C13 enriched. So there would be a twofold decrease in isotopically labeled pyruvates and also a twofold decrease in other isotopically labeled metabolites and amino acids. And because glucose starts at the very beginning of carbohydrate metabolism, we would have much less metabolic control over where it can end up going. So we could get a multitude of amino acids getting labeled, including glycolytic amino acids and citric acid cycle amino acids. And this is just showing you how glucose undergoes glycolysis and eventually the citric acid cycle through pyruvates. So you could get all the amino acids coming through either glycolysis or the citric acid cycle or pyruvate itself. When we use pyruvate, we eliminate all the all the glycolytic amino acids, and better yet, if we use methyl labeled pyruvates, we eliminate a lot of the citric acid cycle amino acids as well. We would be expecting amino acids more along the lines of the aliphatic amino acids, as well as some of the alpha ketoglutarate and oxaloacetate amino acids. So we would be specifically looking for methyl labeling in the longer chain residues like isoleucine, leucine, valine, and obviously as well as alanine, because a simple transamination reaction would give us alanine from pyruvate. And we could also see proton deuteron exchange through that enzyme as well. So now I'm going to talk a little bit about the overall method for how we wanted to do this, and then I'll go a little bit more in depth about each step specifically. So ideally, we would like to have deuterated pyruvate prepared prior to starting the culture. This isn't something we've worked on this semester, but we have worked on in the past. So I'm going to give I don't think I've updated on it. I'm going to give an update on it today and how we're going to approach that maybe possibly next semester as well. Because pyruvate's so reactive, it can be difficult to actually deuterate its methyl carbon. So we're going to essentially grow a culture in LB and then move them into a smaller culture in M9. It's more restrictive of a medium to let the cells get accustomed to the harsh conditions. Cell growth can sometimes be difficult in M9, and growing in a small M9 makes it easier for them to grow in a larger M9 culture. Then around the exponential growth phase, we can add our isotopically labeled pyruvate, and then add our protein inducer for our proteins to get expressed. And then we can purify our protein and analyze it via NMR using a methyl edited pulse sequence and getting those sharp triplet signals that I was talking about. So, like I said in the past, we have used multiple ways of deuterating the pyruvate methyl carbon. We tried an enzyme catalyzed reaction using the alanine aminotransferase enzyme, but the reaction is reversible and wouldn't go to completion. So we would get multiple species of multiple deuterated species. We would see the fully deuterated methyl carbon with the partial deuterated 1 deuteron and the 2 deuteron species, when in reality you would like to have fully deuterated methyl carbon. We also tried a base catalysis for the purpose of deuterating pyruvate, expecting ketoenol tautomerization to undergo, allowing for proton-deuteron exchange. It seemed efficient in the spectrum. The pyruvate methyl would get fully deuterated. However, the highly nucleophilic properties of pyruvate would undergo an aldol condensation, leading to parapyruvate dimer. And this dimer is actually very toxic to the E. coli cells and inhibits a lot of the metabolic function of the cells, which would be very obviously detrimental for our purposes. But then we found this heated reflux method using pyruvic acid, and we tried it using sodium pyruvate in acidic conditions to basically replicate pyruvic acid ourselves. However, the dimer still formed, and it seems like you actually need to use pyruvic acid. You can't just use sodium pyruvate. So that's something we look to do in the future. And again, this isn't something we worked on this semester. So this semester, we started with a cell culture with the intent of expressing protein with isotopic labeling incorporated into it. And that is, of course, through the use of methyl labeled pyruvate. So for our cell culture, we use the common protein expression host, BL21DE3 cell line with kanamycin-resistant plasmid. And for our protein, we use the TgSkp1. It's a good candidate for this project. It's a 20 kilodalton and 40 kilodalton dimer. It's got a highly mobile C-terminal region, which is a glycosylation site and has lots of residues with the pyruvate methyl carbon can potentially incorporate itself into, like alanine, leucine, isoleucine, and glutamate, especially towards the C-terminus there. The protein also has a polyhistidine tag we can use for purification using IMAC purification, and I'll talk a little bit more about that. So our cell culture specifically, we initially grew our cells in a nutrient-rich LB growth medium, and this took about 4 hours. We incubated for 4 hours until the cells had grown to confluency. Then we wanted to transfer these cells to a small M9 culture before going into a larger M9 culture. So we inoculated a 5 mil M9 culture with the cells at 2% of the new culture volume, so 100 microliters, and let this incubate overnight. Once we saw that they'd grown the next morning, we transferred them over to a larger 250 mil culture, again at 2%, and let these grow. During the exponential growth phase, we added our isotopically labeled pyruvate in. This is when the cells are rapidly dividing, so they can make use of the pyruvate. And prior to adding our protein inducer, we waited 20 minutes for the cells to use the isotopic labeling and incorporate it into some of its amino acids. So then 20 minutes after the isotopically labeled pyruvate was added, we added IPTG inducer, dropped the temperature to about 22 degrees Celsius, and then let it express protein for about 16 hours and came back the next morning to collect our samples, ready for them to be purified. So once our protein was expressed and we'd collected our samples, we were ready for purification. We use IMAC purification. It's a kind of affinity chromatography that uses our protein's polyhistidine tag. It works by forming coordination complexes with the histidine residues. And this works well because the histidine compounds have lone pair electrons that can share with the MTP orbital of the metal resin that we use. In this case, we use cobalt. It's divalent metal, so like CO2 plus, and forms a strong bond with the protein's polyhistidine tag, and everything else that doesn't have affinity for the resin will elute through. So we first lysed our cells via sonication and enzymatic lysis using lysozyme. We also use protease inhibitors, lupeptin and aprotein to prevent any protein degradation. Pellet and lysate samples were separated and we aliquoted them for a protein gel. And then we used a gravity column and kept it cold at 4 degrees Celsius the whole time. Equilibrated the column using wash buffer initially and coated it with our cobalt resin. So then the lysate that we had collected from the sonication, we applied the lysate over the column, washed the column again, allowing any protein that wasn't bound to the resin to flow through. The flow through was also aliquoted for our protein gel. And at this point, the SkpOne was bound to the cobalt resin. And we just wanted to elute the protein using imidazole. Imidazole is structurally similar to the histidine side chain, so we can use the compound at high concentrations, allowing it to form coordinate bonds with cobalt instead of histidine. So the imidazole will bind the cobalt, and the polyhistidine tags will unbind and allow the protein to elute. We collected the eluted protein, also aliquoted that for the protein gel, and dialyzed it using a 12 kilodalton dialysis membrane, put it into tris and salt buffer. And then we concentrated the protein using a 10 kilodalton filter, and the final concentration was 380 micromolar. And at this point, like I mentioned, we had taken aliquots at 5 points to make a protein gel, so we can qualitatively assess our protein. And here are those gels. The lanes are listed up there, right up there where it says lane 1 through 5 and what they're loaded with. We made a Kamasi blue and an antihistidine antibody gel, a Western blot. Looking at the first 2 lanes in the Kamasi, loaded with pellet and lysate respectively, we can see that there's no signal here. Here we would expect to see a ladder similar to what we see in lane 3, but the absence of the signal could just be because our load volume was potentially too low. In lane 3, this is where the lysate flow through was loaded. Here we can actually see the ladder coming from everything that wasn't bound to the resin, and we can also see some SkpOne as well at 20 kilodaltons. In lanes 4 and 5, we can see our SkpOne signal with lane 4 was the eluted protein that was captured and eluted with imidazole, and lane 5 was the concentrated protein. And we can see that lane 5 seems to be anywhere from 10 to 20x stronger signal than what's in lane 4. In the Western blot, we can see SkpOne signal in all 5 lanes, with the signal getting stronger from left to right. We can see a small SkpOne signal in the pellet, meaning we collected most of our protein with little remaining in the pellets. And the lysate is the soluble component of the protein. And we also see a fair bit of signal in lane 3, which was the lysate flow through. This indicates that some of the protein had not bound to the resin, and this could be because the protein expressed exceeded the capacity of the resin to collect. So we expressed quite a bit of protein, more than we expected. Lanes 4 and 5, we can see our SkpOne signals. Lane 4 was the eluted SkpOne, and lane 5 was the concentrated SkpOne at 20 kilodaltons. And you can also see the dimer at about 40 and a very, very brief signal of the trimer at around 60. Looking at the SkpOne samples in lanes 5, you can see that there is no other signal absent, indicating the purity of the protein, especially because the protein was concentrated. So any other impurities that were here, or any other substances in the sample, would have also gotten concentrated and shown up here. But the absence of those signals shows high levels of purity. And then we also used the nanodrop to quantitatively assess the SkpOne, and we collected 11 milligrams of protein. So at this point, we had collected our SKP1 protein, and we knew that it was quite pure. Our samples were quite pure. So this helps us, as we know, any signal in the spectrum is coming from the protein. So things we want to know now is how heavily was the isotopic label incorporated, and was the isotopic label even incorporated, and what amino acids was it incorporated into? So I prepared some NMR samples and ran a few NMR experiments, few basic 1D HSQCs as well as 2D HSQCs. And the first spectrum I see is this one. And initially, the first thing you notice, there's quite a bit of signal here. You look downfield of the water signal, and you see no signal present there. So that indicates no aromatic labeling was present. Something we didn't expect anyways, because tryptophan, tyrosine, and phenylalanine are all glycolytic precursors. So that's a promising sign. We look upfield the water signal, and we see that there's some signal in the alpha carbon region, as well as the methyl region, the methylene region, and in between, where we may see some beta carbon labeling of certain amino acids. So in an attempt to potentially see how much isotopic label was incorporated, Dr. Pressegard and John recommended we get some mass spec work done. So Dr. Azadi, I believe is the name, I may be mispronouncing it, their group helped us run the samples through mass spec, and when the results came back, the results had shown that much of the protein was labeled to a great extent, and in many different amino acids. Labeling had been observed in aspartate, asparagine, glutamine, glutamate, lysine, the aliphatics, methionine, threonine, and arginine. And that's a lot of signal. Although these signals are downstream of pyruvate, this is still quite a bit of labeling for just the pyruvate methyl to have incorporated into. So so at this point, we were wondering how this could have possibly happened. How do we have so much labeling? So we did some troubleshooting. And is it possible that we got extra carbon into the samples? I checked the glucose, and the glucose was unlabeled. I then checked the pyruvate stock that I had used. And so we had intended to use the methyl labeled pyruvates from Cambridge Isotopes. I checked the NMR, I check a carbon spectra, and I see 3 peaks in the carbon spectra, one for each of the carbons. And I read the label as well in the fridge, and I see that it's fully labeled pyruvates. So I made the mistake of reading the actual 13 or 3-13C pyruvate, 13C3 pyruvate, rather than just looking up the lot number on Cambridge Isotopes. So obviously something I learned to be more mindful about. I used to keep my pyruvate in that, my like methyl labeled pyruvate in that fridge like 6 months ago, I think last fall, or maybe a year ago. And I definitely should have double checked with Cambridge before putting it in. But we have since actually ordered methyl labeled pyruvates, and I'm making sure storing separately and keeping track of what's what, being more mindful of that. But how this affects our project, or the current protein that we had already expressed, it makes more sense why there was so much signal in the spectrum, and why the mass spec had come back with so much signal. So, because now there's more amino acids that can be labeled, and to a greater extent, it's not only limited to the methyl carbon labeling, but alpha and beta carbon labeling, excuse me, as well. The pyruvate first and second carbon can be incorporated, and the citric acid cycle may scramble the carbons more than they would if it was just a methyl carbon. So what I did at this point, knowing the possible amino acids that pyruvate could incorporate into, I tried to analyze the spectrum a little further, since we knew the pyruvate was fully labeled. And because there wasn't much prior toxoplasma SKP1 NMR data I could use, I ended up using chemical shift data from BMRB. They have histograms of all amino acid chemical shifts with means and standard deviations, which I used to try to assign my own peaks. So I separated the spectrum into 4 distinct regions. Region D down here below is the region, the alpha carbon region. Here we can see isotopic labeling coming from the carbons 1 or 2 of pyruvate, but not the methyl carbon, most likely not. Region C is the methylene region, where pyruvate may have incorporated into amino acids like aspartate, asparagine, or the methyl, particularly glutamate and glutamine, has been seen to be labeled in the past. Region B is where we see a lot of the beta carbons of the amino acids in this region that wouldn't necessarily get labeled by the pyruvate methyl, but more likely to get labeled by the first or second carbon of pyruvate. A lot of the beta carbons in this region are coming from citric acid cycle intermediates. SkpI is particularly abundant in lysine residues and lysine synthesized through condensation of pyruvate and an aspartate derivative, which in this case would both be labeled. So a lot of the labeling here, I suspect, could be coming from lysine carbons, maybe the beta carbon as well. Region A is the methyl region, where we would expect the pyruvate methyl carbon to incorporate itself into some of those aliphatic residues like alanine and longer chain isoleucine, leucine, and valine residues. So from here, I decided, knowing what the initial goal of this project was, and because we had intended on using methyl label pyruvate, I decided to just focus on the regions where the pyruvate methyl may have incorporated into. So the aliphatic amino acids, glutamate potentially, and I believe threonine and methionine as well. So using the proteins sequence and the mass spec data and the NMR data and BMRB, I tried to assign peaks in the spectrum corresponding to these residues. And here I can show you expanded regions of the methyl and methylene. Like I said, high labeling of aliphatic residues expected, and that's what we see here. We can see alanine peaks further downfield from the longer chain residues, and we can see the longer chain residues are closer to each other in regard to chemical shifts, with isoleucine being furthest upfield, and the signals are overlapping a bit. And in the methylene region, we would expect glutamate labeling and as well glutamine, methylene signals. Glutamine is the most abundant amino acid in SkpI, more abundant than any other residue in the protein. So high glutamate labeling is actually expected. And we can see quite a bit of signal and lots of potential signal overlapping here. That could be, it's kind of difficult to quantify how many signals are under that large, broad peak. So we learned a good bit from this semester overall. We learned that our culture conditions for the E. coli cells are good. They have no problem growing after labeled pyruvate addition. We also learned that they may not need as high of a pyruvate concentration as we think. We had used 25 millimolar pyruvate in the past, and this semester was the first time we used 12.5 millimolar. And as you could see, there was still quite a bit of signal. We also learned that SkpI has no problem incorporating the isotopic label, and C13 gets incorporated very well into the protein. We were able to see methyl labeling in the methyl region, where we would expect signal from the aliphatic residues, and we were able to see alanine's peaks well separated from the longer chain residues, which means simultaneous labeling of different methyl groups is possible, which is promising for next semester's experiments. We saw that pyruvate went a lot more into residues than we expected. We had labeling of, since we had labeled all pyruvate carbons mistakenly. So we got to see some citric acid cycle amino acids get labeled, and we observed that glycolytic amino acids didn't get labeled. The aromatic, tryptophan, tyrosine, phenylalanine, as well as serine and histidine. We've learned a fair bit moving forward, so I think we can take a lot away. But moving forward, obviously, we're going to want to do this again using methyl-labeled pyruvates. And we may want to try the heated reflux using pyruvic acid instead of pyruvates. And we could also optimize our protein expression. Like I mentioned earlier, for this expression, our induction was 16 hours. And as we saw in the protein gels, we had collected much more protein than expected, even exceeding the column's capacity. But more importantly, how would a longer induction period affect the isotopic labeling incorporation in our protein? And also maybe it would affect the labeling pattern of the amino acids as well. And something else that could affect this is how long after pyruvate addition we added our IPTG inducer. This time around, it was 20 minutes, but it's something to consider moving to the future. And also seeing that it was the first time we used 12.5 millimolar pyruvate concentration, we still got plenty of signal. Maybe seeing if we can potentially get away with using less pyruvates in the future as well. And yeah, that's it. Thank you.